Okay, welcome once again to this lecture. I'll be discussing to you the part 2 of this topic on enzyme. In the first video, I discussed the processes on how enzymes are named. Okay, now, now this time, I'm gonna be bringing to you the 6 major classes of enzyme. Remember, enzymes, let's review. They are proteins. Okay, most of the enzymes or majority of the enzymes are proteins. Okay, and they are classified based on components, simple and conjugated enzyme. So there are many enzymes and they are classified into 6 major classes. Namely, okay, we have the first oxidoreductase. By looking at the name, all right, reductase or oxidoreductase, it means that this group of enzymes, okay, what they do is to catalyze what? Oxidation and reduction reactions. Okay? So these are group of enzymes that catalyze oxidation and reduction. Okay? Um, they are known to be a redox enzyme. And we know that if it is redox, they are always linked to one another. Okay? Oxidation and reductions are reactions that are always linked to one another. Okay? I will explain that in this reaction. An oxidoreductase requires a coenzyme that is either oxidized or reduced as the substrate in the reaction. Okay, let me explain this redox reaction in this or using this illustration or using this reaction here. The substrate is lactate. Okay, and the enzyme is lactate dehydrogenase. What lactate dehydrogenase does is to catalyze the uh, oxidation, okay, oxidation of lactate. So lactate to pyruvate, this is an oxidation. How do we know that this process is oxidation? Okay, take note that you have here an alcohol, a secondary alcohol. Remember, I said before in my previous videos that if you have a secondary alcohol, what happens is when it's oxidized, it becomes a ketone. Okay? Remember, take note that a secondary alcohol, when it's oxidized, it becomes a ketone. So, the reaction is oxidation with respect to lactate being converted to pyruvate. Alright? Now, what is the role of this? Okay? What is the role of this molecule here? NAD serves as oxidizing agent. Okay, let's just write here OXA, oxidizing agent. What is an oxidizing agent? All right, oxidizing agent oxidizes the lactate. Okay, the oxidizing agent oxidizes the lactate or any particle. Now, what catalyzes the oxidation? It's the enzyme lactate dehydrogenase. So they are different. The oxidizing agent is um, it's the one that oxidizes the lactate. in order to facilitate the oxidation, okay, um, in order to increase the rate of oxidation, you need an enzyme. Okay? So that is the work of enzyme and the NAD. NAD plus. NAD plus, this stands for, okay, nicotinamide. The N is nicotinamide. This is derived from a vitamin nicotinic acid. A stands for adenine. Okay, adenine. And D stands for dinucleotide. Okay, adenine dinucleotide. Nicotinamide adenine dinucleotide. What does the plus mean? Okay, the positively charged. Okay, the positively charged indicates that the NAD is in the oxidized form. Okay, it's in the oxidized form. Okay, so I repeat, the function of NAD is to oxidize the lactate. Okay, the lactate is oxidized to pyruvate. That's why it's called oxidation. Now, take note what happens to the oxidizing agent. What is our oxidizing agent? The NAD. Take note that the NAD becomes NADH. Okay? NADH still stands for nicotinamide adenine dinucleotide, but the H represents hydrogen, meaning the NAD has been, okay, the NADH is now, okay, in the reduced form. Okay, NADH, that means it's a nicotinamide adenine dinucleotide in the reduced form. Okay? I mentioned that redox reactions are always linked to one another. Okay? So how do the reactions that we have seen earlier link to one another? So you have lactate. Okay? You have lactate that has been oxidized by the NAD catalyzed by the enzyme dehydrogenase. Okay, let's just put here dehydrogenase. And the lactate has been oxidized to pyruvate. Okay, so where is the oxidation? The oxidation process is the lactate to pyruvate. And where is the redox? Or where is the reduction, rather? Where is the reduction? The reduction is the NAD plus being converted into NADH. As I said earlier, the NAD plus is in the oxidized form and the NADH is in the reduced form. Okay? What is the role of the NAD plus? It serves as oxidizing agent because it's in the oxidized form. You cannot function as an oxidizing agent if you are not in the oxidized form. Okay? So any oxidizing agent, all right? Any oxidizing agent gets reduced. That's why here in the reaction, you notice that the NAD plus, which is an oxidizing agent, becomes reduced to NADH. That's why in the previous slide, I said that redox reactions are always go together. Any oxidizing agent gets reduced, and any reducing agent, okay, if you have here a reducing agent, okay, a reducing agent gets oxidized. Okay, the second group of enzyme, the second major class of enzyme is called transferase. That means these enzymes, the enzymes that belong to this class, what they do is to transfer functional group from one molecule to another. Okay, to transfer. There are two major subtypes. Okay, the following are transaminases. What they do is to transfer amino group, and the other one is kinase or kinases. What they do is to transfer phosphate group from specifically from ATP to a substrate. Okay? So transaminases transfer amino group, and kinases transfer phosphate. The source of phosphate is ATP. Okay? From ATP, remember there are three phosphate groups here in ATP. One of the phosphates is transferred to a substrate. Okay? So for example, here in this reaction, the substrate is fructose 6-phosphate, and then you have here ATP or adenosine triphosphate. All right, the enzyme phosphofructokinase brings or attaches one of the phosphates of ATP, and it's added to the substrate. Okay, and you notice the product becomes what? Fructose 1,6-bisphosphate. There are now two phosphate groups on carbon number 1 and carbon number 6. Notice what happens to the ATP. It now has only two phosphate, so you call that adenosine diphosphate or simply ADP. The third class of enzyme, the third major class of enzyme is called hydrolase. I mentioned earlier that when you say hydrolase, these are enzymes that catalyze breaking of bond in the presence of water. So they catalyze hydrolysis reaction. There are many enzymes that belong to this, and they are further classified based on the reaction that they catalyze. Let's say, for example, carbohydrases, they break or they hydrolyze glycosidic bond in carbohydrates. Proteases, okay, they are enzymes that belong to the hydrolysis of protein or breaking of the peptide bonds. And I've said earlier, lipase or lipases, they are involved in the breaking of the ester linkages in triglycerol. Remember, triglycerol is built from the reaction of glycerol and three fatty acids. And in triglycerol, you have three ester linkages. So the three ester linkages are hydrolyzed by lipase. So here you have a polypeptide in the presence of water catalyzed by an enzyme protease. Protease is, as I said, it's still a general term. Okay? So what happens here is the peptide bond is broken. The next group is liase. Okay. Now this group of enzymes, what they do is to catalyze addition of a group specifically to a double bond. Okay, or removal of a group to form a double bond. So for example, you have here fumarate. Okay, the functional group here that we want to consider is the double bond. Okay, so this is unsaturated. So when water is added to the double bond, okay, you will form a secondary alcohol. That's the forward reaction. However, when um, when the water that has been added is removed, you will form a double bond. Okay? Take note that in liase, the water molecule is part of the molecule. Okay? The water, the atoms in water becomes part of the molecule. Unlike in the hydrolase, water participates in the breaking. Water does not become a part of the molecule. So here in the fumarate, the water molecule or the atoms in the water become part of the malate. Now, when malate is when malate undergoes removal of water, okay, here water is released, okay, water is released, denoted by this arrow, okay, it will go back to fumarate, which is you have an alkene or you have a double bond. All right. The fifth class is isomerase. What are enzymes? What do they do? What are these enzymes up to? Isomerase, these are enzymes that catalyze isomerization, or simply the word isomerization is rearrangement of atom. So basically, what you have here is, for example, you have a ketone by the name of this molecule, dihydroxyacetone phosphate. Okay? When this rearranges, this becomes a secondary alcohol. So if you will count the number of atoms, you will find the same. Same number of carbons, same number of hydrogens, same number of oxygen, and you have the same number of phosphorus. So basically, rearrangement, no, rearrangement of the structure, but the chemical formula or the molecular formula is the same, but the structure differs. And the sixth, sixth and the last group is called ligase. Ligase, ligase is a group of enzyme that catalyzes the formation of new bond or formation of a bond between two molecules. Okay, so you form a bond by combining, okay, two molecules. Okay, here what being combined are the following: pyruvate and carbon dioxide. Okay, so carbon dioxide becomes part of the molecule. So here you have oxaloacetate. So this portion here uh, is the carbon dioxide molecule, but we don't recognize this as carbon dioxide because it's now part of the molecule. Okay, if it is CO2, then that's carbon dioxide, but it's now part of the molecule, so the whole molecule is called oxaloacetate. So in order to form a bond, it requires energy and the source of energy is from the hydrolysis or from the breaking down of ATP. So take note that ligase, we are forming a new molecule by combining two, okay, in the presence of energy from ATP.